Hello friends. So in this week we will cover bioaerosols in the indoor atmosphere. So bioaerosols are essentially tiny airborne particles that are biological in nature. So these are like aerosols but biological in nature. So we had physical, chemical aerosols. These are bioaerosols. These are the particles that are derived from biological organisms. And they may include viruses. So just a sketch of virus here. They may include bacteria, fungi and fungal spores, pollens, and also the various fragments of animals and the plant organisms. So anything that is derived from biological material or biological organisms can be termed as bioaerosols. These include the body parts and feces of insects such as dust mites, cockroaches and other insects. So the insects themselves are very small and the body parts and pieces will be even smaller. So if they are on the floor and somehow they or other surfaces, and when the dust gets resuspended, these biological parts will also get resuspended, and once they come in the air, there is a chance of inhalation by humans. Bioaerosols would also include the proteins that exist in saliva, urine, and dander of furred animals, cats and dogs. So also the animal related material could be in the suspension in air as bioaerosols. Bioaerosols include also endotoxins. Endotoxins are basically the main component of the outer cell wall of gram-negative bacteria. So these are basically parts from the bacteria. Then also include beta-glucans from cell walls of bacteria, fungi, yeasts, algae and also some plants. They can also include, the bioaerosols can also include low molecular weight secondary metabolites. For example, the mycotoxins. Bioaerosols may also include antigens and allergens. Bioaerosols can also include microbial volatile organic compounds, so MVOCs. This is a sort of an illustration on what humans can give and humans can be exposed to. So, one can be exposed to fungi, bacteria, mycotoxin, different viruses, and viruses we already know through the famous COVID-19, but in our everyday life as well, we may be exposed to different fungi and viruses. Then we have allergens, so more like plant, insect and animal based substances inhaled by a human, and the endotoxins. So, and this exposure may be occurring almost all the time or most of the time. Sometimes this may lead to harmful effects, and these effects could be things such as allergic rhinitis, asthma, watery eyes, headache, coughing, again allergy, dizziness, fever, legionellosis and tuberculosis, TB. So all these kind of bioaerosol exposure may be happening in our everyday lives in a way. And sometimes we will be okay. Sometimes if we have acute exposure, we may inhale some particularly dangerous virus, then we get infected very quickly. And sometimes these exposures are chronic in that we are exposed to low dose but over a long time, and will develop health effects over a long term, but then they will be there. So all kinds of exposures are possible, and these are some of the health impacts that may be possible. So it's important to define what is a biological agent, because we are interested in bioaerosols and the exposure. So a biological agent can be defined as a substance which is of one, biological origin, because we are talking about biological agent, so it must be of biological origin, and it is capable of producing an adverse health effect. So biological nature, exposure of something which is biological in nature, and then the leading of an adverse health effect. Such biological agents are quite ubiquitous in nature. They are everywhere because we are surrounded by biological things. These biological agents may be, almost everywhere in nature. And the impacts or their influence on their origins and their abundance can be amplified in man-made environments, because we may provide conditions that either make them more potent or make them more in concentration, and these agents can thus be amplified in man-made environments and in the materials. This is an illustration of an order of magnitude of the viability or the persistence of microbial particles in air and on surface. So this is our biological agent. This is the sort of persistence of viability in the air itself, and this is the viability or persistence in a surface. First we will look at the influenza virus. In the air, their viability is up to 21 days, but it decreases to up to 11 days when it is on a surface. If we look at staphylococci, it is the opposite direction. So in influenza virus, we have potentially more persistence in air and a bit lower on the surface. When we look at staphylococci, the trend is the opposite. In the air, they may be viable or persistent up to about 3 days. But when they are on surface, they may be viable or persistent from 7 days to up to 7 months. So this is a very long duration. Same goes for streptococci. While in the air, they may be viable or persistent for up to 48 hours, which is 2 days. On surfaces, they may be viable or persistent from 1 day to 6.5 months. So again, a very long duration, they can be remaining viable in the surface. Same for E. coli. While in the air, the viability is around 60 minutes, that is around 1 hour. In surfaces, somehow they can survive more, from 1.5 hours, which is 90 minutes, to even as much as 16 months if the conditions are very conducive. Legionella pneumophila, its viability in air is 15 minutes, but it is not detected on surface. So, one, it is not very persistent and probably not at all on surfaces. Then we have Aspergillus and Penicillium conidia, which in the air and in the surface, on both media, it can be surviving up till 22 years. So this is a very, very long time. Its survival is very long. So therefore, the impact could be long term. Then the dust mite allergens are there. In the air, they may survive up to few months, and same on the surface. So here we see different kind of biological agents have very different viability time span. Aspergillus and Penicillium conidia can live up to 22 years, which is a very long time. Dust mite allergens few months, and both in air and surface. For the influenza virus, we see that viability or persistence in the air is somewhat larger than on surface. But when we look at Staphylococci, Streptococci and E. coli, the viability in air is from 1 hour to 2 days to 3 days, but on the surface it goes from 1.5 hours to 7 days to up to like 6 to 16 months. So many different biological agents have very different viability or persistence depending on if they are in air or if they are in surface. So their characteristics are different than each other, and even their interaction with the environment are different. If we look at the size ranges of different individual bioaerosols, fragments can go very low. So here they have gone to 0.001 nanometers, 0.001 micrometers, so 1 nanometers. And so we will start from pollen. So pollen can go around 100 to 10 micrometer size range. Fungal spores, which are here, can be between around 30 to 2 micrometer size ranges. The microalgae can be from 1 micrometers to more than 100 micrometers. Then the bacteria can range from 0.3 micrometers to more than 10 micrometers. So some of the bacteria, as we say that anything above 10 can settle out fast, the pollen may not be suspended in air for a long time. Some of the fungal spores will be suspended, some of the microalgae will be suspended. Also, quite a bit of bacteria will remain suspended because their size ranges are 0.2 to around 10 micrometer. Same goes for viruses. The viruses size range is around 0.2 micrometers to 0.02 micrometers to 0.3 micrometers. Then we have the fragments of all these, which can go from around 3-4 micrometer to very low numbers to the order of nanometers. These are the typical size ranges. A protein can be around 1 nanometer size range. A virus can be around 50 nanometer size range. A bacteria is around 500 nanometer here. A fungal spore around 1 micrometer. So this is a fungal spore. And then a pollen grain could be around 5 micrometers. This is another illustration. So Mycobacterium tuberculosis is around 2 micrometers. So there is a chance it will be suspended in air. Flavivirus is 50 nanometers or 0.05 micrometer. The redbud pollen here is around order of several tens of micrometers. And Chlamydomonas, which is about green microalgae, is of the order of 10 micrometers. So they come in very varying size ranges. The next is, we will again look at this from different scales, from larger to smaller. So we have the frog egg, which is around 1 millimeter in size and can be looked through a light microscope. So on the x-axis we have different size ranges. then we have different materials and the method in which we can use to detect them. So our frog egg is 1 millimeter and detected through a light microscope. Then human egg and pollen are around several hundred micrometers. Then we have the plant cell and the animal cell, and they are somewhere between 10 micrometer and 100 micrometer. It's in log, the scale is log scale, so I think these are around 50 micrometers or 30 to 50 micrometer sizes. Then we have the red blood cell, which is around 7 to 8 micrometer size. And these can all be looked through light microscope. Then we come further down and we have the bacteria and mitochondria around 1 micrometer size range. Then we go to submicrons now. So smallpox virus is around 0.5 micrometer, and this is sort of the size range. When we recall the week on aerosols, this is sort of the size range which is quite likely to be suspended in the air. Or some of the lower size range can go nucleation and come in the accumulation mode particle size range. Then we have the flu virus, 100 nanometer or 0.1 micrometer. These things can also be looked through an electron microscope. The polio virus is around 20 to 30 nanometers, so 0.02 to 0.03 micrometer right here. Then we have the proteins, which are around several nanometers. Same for lipids. A C60, which is a fullerene molecule for comparison, is 1 nanometers, and all can be looked through an electron microscope. Finally, our atom, which is on the scale of angstroms or 10 to power minus 10 meter. So this is 0.1 nanometer. So we are looking at different size scales. So our proteins are maybe around 50 to 100 times bigger than atom, and the polio virus would be around 200 times bigger than the atom. But we see what are the different size ranges at which our biological particles and the bioaerosols would be here. Again, this is another way of illustrating. So the atom is 0.1 nanometer, DNA is around 1 nanometer, our viruses, bacteriophages will be around 10 to 100, bacteria around 1 micrometer, then human cell is around here, and the red blood cell around 10 micrometer. As a comparison, there are 10 to the power 13 human cells in the body, 10 to the power 14 microbial cells, 10 to the power 12 bacteriophages in 1 gram of fecal material, 10 to the power 11 bacteria in 1 gram of fecal material. So we are just trying to get a magnitude of the scale. Now bacteria, we will come to bacteria. Bacteria are unicellular prokaryotic microorganisms. These are found in almost all environments on the earth. Compared to the number of cells in the body, humans have 10 times more bacteria on their skin and their gut flora. Although airborne bacteria can travel in single cells, they usually cling to other particles. So as single entities basically, they can usually cling to other particles like soil, leaves or skin flakes or pieces of skin. The shapes of bacteria can be of different types. We can have spherical bacteria, which are these cocci, almost spherical shape. Example could be Micrococcus or Streptococcus. They can often occur in pairs and chains and clusters. Then we may have the rod-shaped bacteria. So like this, rod-shaped bacteria. For example, the bacilli, example being Bacillus anthracis, and they may be single or they may form chains. Then we have the spirilla, which include Campylobacter jejuni, Helicobacter, Spirillum vinogradeschi, and these are sort of spiral in shapes. And they range from gently curved shape to a corkscrew-like spiral. Based on the structure of the cell wall, most bacteria can be grouped in two parts. One is the gram-positive or the gram-negative bacteria. Generally, the cell walls of gram-positive bacteria have a thick layer of peptidoglycan. But the gram-negative bacteria will have several thin layers of the same peptidoglycan. These layers are surrounded by an outer membrane coating, which are the lipopolysaccharides, which are also the endotoxins. Some gram-positive bacteria can develop endospores, Bacillus and Clostridium. These are some dormant forms with the following factors such as resistance to radiation. So when we say resistance to radiation, we have to think about air pollution control devices. So if there is equipment which says they will disinfect the air by killing the bacteria, we already should know that for some of the bacteria this may not work as well because they are resistant to radiation. So we need to maybe increase the lamp power or change the radiation wavelength. These bacteria, these forms may be also resistant to heat and also resistant to fleeting and maybe other types of environmental stresses. So these would become very persistent bacteria, very difficult to destroy by radiation, by heat, by freezing, and also other types of environmental stresses. In general, endospores can be both smaller and larger than the vegetative cell. These are some classifications and properties of the gram-positive bacteria. So we will start with the branching filaments. These can be aerobic, nocardia, or anaerobic, actinomyces. Then we have cocci. These are aerobic, examples being Streptococcus and Staphylococcus. This Streptococcus may have partial hemolysis or complete or no, and accordingly they will be alpha, beta and gamma, and then they may have different growth in different media or different sensitivity to different agents. Same for Staphylococcus, whether they coagulate or do not, whether they whether expressed by coagulase or not, and the sensitivity. Then bacilli can be aerobic, which will include Listeria, Bacillus, Corynebacterium, or anaerobic, which would be Clostridium. This is the same type of classification of the gram-negative bacteria. So we will start from here. Diplococci, which would be aerobic, and depending on whether there is maltose utilization, there would be N. norhuia, moraxilla, or N. meningitidis. Cocobacilli could have been H. influenza examples, B. pertussis, Pasteurella, Brucella, F. tularensis. The bacilli could be lactose fermenting or not. If yes, if they are slow, then Citrobacter, Serratia. If they are lactose fermenting and this is fast, we have E. coli, Enterobacter, Klebsiella. If they are not lactose fermenting, what about the oxidase activity? So Pseudomonas or H2S producing? Yes, Salmonella proteus, no, Shigella yersinia. Then we have comma shaped and oxidase positive, which may grow in at some temperature. So C. jejuni, which may grow in alkali media. So this is our V. cholera. And if they are urease producing, so H. pylori. So a lot of classifications are there, and these are broad classifications based on the properties. Bacterial strains, which are generally found in the indoor atmosphere, are representative of the genera Bacillus, Micrococcus, Cocuria, Staphylococcus in general. Of course, there are other strains also possible to be detected, and indeed they are detected. So these strains may be more abundant. This is a chart, and we do not need to go in very details, but this is a chart of the relative abundances of such microbes. And we see that, Pseudomonas is quite common indoors as well as outdoors, followed by Staphylococcus, which are much more in indoors, Acinetobacter, Corynebacterium, this green color, then Roseomonas, and then Micrococcus. These appear to be the major bacteria. But if we see the list, a lot of bacteria have been identified which are present in very different amounts in different locations. We also have different fungi. So Cladosporium, Penicillium, which seem to be quite major indoors and outdoors, and then the other fungi. So in terms of detection, we have a number of genus that can be detected, and they are present in variable amounts indoors and outdoors. Bacilli can produce spores which are distinguished by their resilience to challenging environmental circumstances such as the UV radiation. These bacilli and bacteria can be stressed by desiccation, lack of nutrients, and extreme temperatures. So these are basically the bacilli are quite resistant bacteria, as we have seen earlier, and their spores that they produce, they are resistant to very much challenging conditions like UV radiation, and they are resistant to desiccation, resistant to lack of nutrients also, and resistant to extreme temperatures, hot or cold. So these kind of spores can survive very well in the environment because they are resistant to radiation, water stress, lack of nutrients, and also high and low extreme temperatures. So quite resistant and persistent spores. Now we have the fungi. Fungi are among the most common organisms on earth, and they include molds, rusts, smuts, yeasts, mildews, and mushrooms. Fungi play a very key role as decomposers of matter or biological matter. Fungi for us are also a source of problems. because they can cause crop disease, infectious diseases or allergies. So in general they may be problem when not managed, but at the same time these fungi can be beneficial if we target them, for example when we use them as biological pesticides. So we may ask them to target certain pests. We may have fungi that will target specific pests which are causing damage to the crops, and in that case they will become biological pesticides. Fungi can also be beneficial for the production of antibiotics. The fungi can be classified as yeasts, which are round or oval unicellular organisms such as Cryptococcus neoformans, or yeast-like fungi, which are partly as round, oval yeasts and partly as chains of cells forming pseudomycelium, for example Candida albicans. Fungi can also be these kind of molds which are filamentous fungi. These are true mycelia with aerial and vegetative mycelia, example being Penicillium aspergillus, Mucor and Rhizopus. Then we may have dimorphic fungi which may exist as yeast or filaments, example Histoplasma capsulatum and Blastomyces. Some fungi are unicellular, but usually they may contain multicellular tubular filaments called hyphae. So if we see on the right, this side of figure, this is like a fungi and these are our hyphae, these tubular filaments. Here, basically everywhere. This is a mycelium, and there are a lot of spore producing structures within this fungi. So spores may be produced and then released. And this is being the reproductive structure. It's the spores producing and then reproducing structure. In terms of scale, we are looking at orders of 60 micrometers, this kind of distances. These are the magnification of the central sections, and we are looking at scales of 2 micrometers to 30 micrometers. These hyphae will branch into a network called mycelium. So we can see these networks quite clearly in this figure, and may develop the reproductive spores. Here. It is generally assumed that spores constitute the bulk of bioaerosols produced from fungi. These are found in the size range 1 to 50 micrometers, but more typically between 2 and 10 micrometers. The frequently encountered genera in the atmosphere are Cladosporium, Alternaria, and the actively released Ascospores and Basidiospores. It has been shown that the emissions of the spores may be accompanied by significant amounts of fungal fragments also. So in addition to spores, we may have fungal fragments being emitted to the air. Some fungi can produce mycotoxins, and these mycotoxins are secondary metabolites, and these are associated with acute health effects, meaning sort of immediate health effects after exposure, or high exposure and immediate health effect, and also chronic health effects, meaning effects that develop over a long period of time. Fungal spores are coated with ergosterol and sometimes sugar alcohols such as arabitol and mannitol. Now we have fungal strains, and these are of importance in indoor air samples, which are comprised of like the genera Alternaria, Aspergillus, Cladosporium, Penicillium. These are commonly found in lot of indoor air bioaerosol studies. These are sort of giving us an indication of the size of the fungal strain. So we have the conidia of Penicillium or Aspergillus next to a dry droplet of inorganic salt, and here embedded in a cloud of carbon nanoparticles. Almost all fungal strains are able to form spores, and these spores can be resistant to the changing environmental conditions such as desiccation, availability of nutrients, radiation, extreme heat and extreme cold. So they can be quite resilient. Next we have the viruses. So viruses are parasitic infectious agents that only can reproduce in living cells of animals, plants or bacteria. Viruses are one of the most common biological entities in ecosystems. So we have like a viral particle that can be released from a cell and can be capable of infecting other cells, and this is a virion. All viruses carry nucleic acid either as DNA or RNA, but not both. A protein shell called a capsid can surround the genetic material, which is the source of infection. The capsid allows for recognition of receptors on the host cell and helps the virion in some cases to enter the host cell. So this is the virion, the viral particle released from a cell and capable of infecting other cells. So capsid allows recognition of receptors on host cells and then helps the virion in some cases to enter the host cell. In addition, the viruses can sometimes develop an envelope of lipoproteins originating from the cell membrane. We can have a comparison here. So we have the human cell here 10 to 30 micrometer, bacterium 2 to 3 micrometers. We have picornavirus which is 20 to 30 nanometer, herpes virus 160 to 230 nanometer, basically 0.16 to 0.23 micrometers. This one 0.02 to 0.03 micrometers, so much smaller than the human cell. The HIV virus 100 to 130 nanometer, meaning 0.1 to 0.13 micrometers. And the influenza virus, which is 80 to 120 nanometer or 0.08 to 0.12 micrometers. So all of these are substantially smaller than the human cell. If we look at the viral architecture, as we see, we have the nucleic acid genome here, which is surrounded by the capsid proteins, nucleocapsid, which is capsid and the nucleoacid. So viral capsid proteins protect the genome and are composed of nucleic acid. Capsid and nucleic acid then are known as nucleocapsid, and this capsid protects the actual genomic material, the genome. This is a naked virus, and these are the virus attachment proteins, these ones. So when we have the enveloped virus, the lipid membrane derived from the cell wraps the capsid of an enveloped virus. So this is the envelope now. This was our capsid, and this is the lipid membrane derived from cell which has wrapped. This here is the matrix protein. Now binding or docking of the virus to the host cell is facilitated by this virus attachment protein. So this docking is facilitated by the virus protein, and then the virus can gain entry to the cell. This is a helical capsid structure for information, where viral capsid proteins form the helical nucleocapsid by winding around the nucleic acid. The main source of indoor viruses, once the human is infected, of course, with the virus, is human being. About 60% of total human respiratory and gastrointestinal infections can sometimes cause morbidity in case of extreme exposure or exposure to very harmful virus. In general, a decline in quality of life and also a loss in productivity. This is how a virus may sometimes spread indoors. We can start with the infected individual who shreds virus by sneezing or through elimination of fecal material. So infectious virus transmission can involve combination of both respiratory and fecal oral routes. So the virus is shed from the mouth. When it is shed on the fecal form, and when the toilet is flushed, aerosols will be formed. Now droplets greater than 100 micrometer can travel maybe around 1 meter or lower and fall in the ground within 5 seconds or so. So maybe they cannot be inhaled. So let us say these ones, they went and are dropped within 1 meter. But the aerosol which are pathogens and viruses less than 5 micrometer and let's say 5 to 100 micrometers, which is the yellow color, and 5 micrometers are these very small dots. They can go beyond 1 meters and can stay inhaled. So now they are going beyond 1 meters, maybe 5 to 100 micrometer may still go down a bit sooner, but the ones which are less than 5 micrometers can remain quite suspended and go on the surface. Once on the surface, we can also have resuspension due to different activities. So re-aerosolization from the contaminated environmental surface. Then one can have transmission to other people. Well, one can have transmission through other people through this re-aerosolization or resuspension of aerosols. So let us say this is indirect transmission. Of course, the direct transmission would be that the aerosol did not settle and went through, and it's transported directly to people because the infected person was very close. So the larger particles are directly inhaled, the smaller particles are settled and resuspended and inhaled. Also, the PM 2.5, for example, can be transported via air and this exposing the other individuals. Then we have the pollens, which is generally produced by plants and then transfer to the male gametes, to the pistils or female structures where fertilization takes place. So they are emitted and generally try to find receptor. The size range of the pollen grains is approximately 10 to 100 micrometers. These pollen grains vary in size, surface structure, densities, and their shapes as well. Normally speaking, pollens have a hard shell which protects the seed and also prevents the deterioration of the genetic material which is present in the nuclei. So this hard shell protects the actual genetic material of the pollen. At high humidity or other environmental circumstances, the shell may rupture into smaller fragments between 0.1 and 5 micrometers. So the larger pollen, which was 10 to 100 micrometer, may rupture into smaller fragments between 0.1 and 5 micrometer, and in this size range they can travel further in air. So we can see here the pollen are generally in the size fraction above 10 microns, but then of course when they fragment they can arrive at these size ranges, which is close to our PM 2.5, PM 10 size ranges, and can lead to airborne suspension. Why? So we have looked at these bioaerosols. What is the concern? One, these are ubiquitous in the environment. They are available or found almost everywhere. They can cause up to 5 to 34% of indoor air pollution. Because of their sizes, major portions of bioaerosols are susceptible to reach the deeper parts of the respiratory system. The particles accumulated in the lungs then can be easily transferred to the blood and then spread throughout the human body. So in this way, exposure can lead to infection. Airborne transmission of viruses, of course we know, has been involved in the spread of many diseases such as measles, mumps, chickenpox, the severe acute respiratory syndrome, SARS, and of course we know the COVID-19. A range of airborne bacteria have been reported to contribute to the spread of disease, such as Streptococcus pneumoniae, non-tuberculosis mycobacteria, Staphylococcus aureus, Mycobacterium tuberculosis, and Bordetella pertussis. So in summary, we have obtained an overview of various microbes in indoor aerosols, that is the bioaerosols, which include bacteria, fungi, and the viruses.